Hi, everyone. We are very happy to welcome you all to the first HCA Town Hall of 2025. We've got a packed agenda ahead with many important updates and presentations. Before we dive into today's program, we'd like to take a moment to reflect on the milestones we've achieved together over the past year. It's been another remarkable year for the human cell atlas community, and that's thanks to your dedication and to your contributions. So most importantly, we want to express our heartfelt gratitude to each and every one of you, the HCA community. Your expertise and your collaborative spirit have driven our progress, and we are incredibly proud of what we've accomplished together. So let's get started. We have an exciting agenda lined up today, and we look forward to engaging with you all throughout the session. Please submit your questions as they come up by typing them in the Q&A section of your Zoom screens, and we'll try to answer as many of them as we can as we go along and at the end of the town hall. So in the last months of 2024, 2 more bionetworks completed their initial atlases. The organoid bionetwork created atlases of neural organoids and of endoderm-derived tissues, and the eye bionetwork created a retina atlas. You'll hear more about the retina atlas today from Dr. Chen, the eye bionetwork coordinator, who's joining us for this session. So we're very lucky and delighted to have him here. Looking ahead, over the next 6 months, we aim to have draft atlases from several more HCA bionetworks, so that by the time we meet in Singapore this summer for the HCA General Meeting in 2025 in Singapore, our goal is to have reached the midpoint of assembling version 1 of the HCA Atlas, with draft atlases from 9 out of 18 biological networks. So we have 50% of our bionetwork atlases completed by mid-year. So in November, we were very excited to reveal a major milestone, the publication of over 40 papers in Nature and associated journals in Nature Publishing Group as part of a landmark human cell atlas collection, a fantastic bundle of papers. These papers, and you can see the beautiful covers here that were designed by members of the community, and they're really beautiful. We're absolutely delighted. So the papers broadly grouped into 3 categories that you can see here on the landing page: analytical methods, developmental biology, and atlas integration, of course, that we'll come on to later today in the session. And it also includes a paper from the HCA Ethics Working Group presenting a data sharing ethics toolkit, and a paper from the Equity Working Group explaining HCA's commitment to building an atlas that represents and benefits all of humanity. We encourage you to explore the full collection on the Nature website on the landing page, which also includes an editorial, an immersive feature article, all research articles and perspectives, and a news and views piece. And I'll hand over to Aviv now.
Great. Thanks, Sarah. So first of all, the atlas, the publication bundle had a really amazing pickup both in the science and in the general media. You can see everything from like BBC News to The Economist reported on it. And that was exciting. I think people now appreciate very much both what the atlas is and why it is important to do it. But one of the questions you get a lot after and during a publication bundle, and definitely one of the questions that journalists ask is, are you done, or when will you be done, or what is still needed in order to be done? And so it's kind of useful, if we go to the next slide, please, to think a little bit about where are we headed. And we see 3 major areas of focus. The first one is in the Healthy Atlas. It is still true that a vast amount of what we've done in the Healthy Atlas is centered on single-cell genomics and especially on single-cell RNA-seq. But of course, in the last few years, the Atlas has really transitioned to other levels, to a multi-omics level at the single-cell level, including chromatin and proteins and metabolites and so on, and also to the spatial level. The second dimension, if you can still go back to the previous slide, the second dimension is the dimension of human diversity, looking across different individuals. The atlas is diverse from day one because, of course, scientists have been doing the studies in many different places, but now we have the opportunity to really get to proper human diversity scale. And the third dimension is in the application to disease. And so the atlas has always been envisioned as a reference to advance our understanding of human disease. And we have seen this across many different studies. So if we go to the next slide, a few of the illustrative examples, really a handful, that have really shown in the last few years were, of course, the big effort on COVID-19, including a recent study in the past year on fatal COVID-19 in the African population, enhancing the understanding of neurodegenerative diseases that really requires comparing post-mortem brain from individuals that have passed from, for example, Alzheimer's disease to the brain reference atlas, which allowed now to develop an understanding of the cells that are implicated at different steps in the progression of neurodegenerative disease like Alzheimer's. Studies into inflammatory bowel disease, both the common form, the common complex form, and the rare form, to identify the cells where the genetic variants that are associated with disease are active, and even helping with generating the necessary predictions of manipulations that would be needed in order to make better cell therapies, which really takes us not just from understanding of disease, but actually to treatment of disease. And so, in the next slide, please, if we start envisioning what the next 5 or so years, 5 to 6 years are going to look like, here are the 4 major things on our roadmap. We have to complete the first version of the atlas. This includes both completing the integration efforts and the annotation efforts, and we're going to hear about examples like those today. We have to build the atlas from its first version, which is cell-centric, into a spatial 3-dimensional atlas. This will include a major data collection effort that, of course, some of it has already been done, but we need to put it really on steroids, and we'll hear about that a little bit today. We need to make sure that the atlas is globally representative, both in the scientists that do it and in the samples that we study. And in order for the atlas really to have its impact, we need the necessary algorithms and platforms and portals so that we can query it to the max, obtain the information that we need, and provide all of that to the many, many different types of users: biologists, computer scientists, physicians, and even the general public through different types of tools and of portals. And so with this, I will hand it over to, in the next slide, I will hand over to John Randell, who will start talking through some of these components with the progress towards the first draft atlas. John.
Thank you very much, Aviv. As Aviv and Sarah have already mentioned, we'll have some really nice detail into both the progress towards the first draft as well as what lies beyond the first draft from Chen and then Chloe Villani and Shyam Prabhakar. In a few minutes, Ray is going to really take you through the iBio Network's approach to Atlas development and how that network has worked with various elements of the HCA data ecosystem to make sure that all the information is posted, and also worked with the organizing committee to meet the criteria for HCA atlases. So I actually want to recap quickly what Aviv and Sarah have already told you about the progress towards the first draft, but really in particular, I want to give you a bit of a window into what it means for an atlas to be approved by the HCA organizing committee as an official HCA atlas. So if we go to the next slide, you've already seen this information once, but as Aviv and Sarah have said, 4 bionetworks have already published their first atlases. Those are the lung, nervous system, eye, and organoid networks. The organizing committee is actually meeting next week, and they will be reviewing the initial atlases from 3 additional networks, gut, liver, and immune. And so we're very excited about that. I will say that typically the organizing committee, which consists of around 36 scientists from around the world, and really is the governance, the scientific governance body for HCA, they will typically hear an initial presentation at one meeting, and then at the next meeting will review how the feedback has been incorporated, and then it'll progress to approval. So we're looking forward to that discussion both at next week's meeting and then the next OC meeting to follow. And then as you see here, the rest of the bionetworks, their atlases are under development. As you've already heard, our goal is to have official atlases from 9 of the 18 bionetworks by the June meeting this year in Singapore, and then to have the rest of the networks completed by 2026. So that is the general timeline. On the next slide, you'll see the general guidelines for what it means to be a V1 atlas in the context of the Human Cell Atlas. So there are really 5 main categories of criteria that an atlas must meet. It has to be robust to technical factors, it has to incorporate previous landmark studies from the field. It really needs to be driven through consultation with the entire biological network at hand. It needs to be annotated with expert input, and then all of the information really needs to be on both the HCA cell annotation platform, which you'll hear much about in this meeting, as well as the HCA data repository and cell by gene. And Ray will sort of take you through that process and the rationale and what it really means from a practical perspective. And then finally, the data, the metadata, and the code all have to be publicly available as well. And on the next slide, you'll see the sort of snapshot that the bionetworks are asked to prepare for the organizing committee. And this is actually the sort of high-level snapshot that's provided to the organizing committee, but then the biological network coordinators will actually present to the organizing committee so that the organizing committee has a chance to ask questions and provide feedback. And I should say that both this snapshot and the presentation are done jointly with the sort of core HCA computational team, which is led by Malte Lücken at Helmholtz Munich, and about a dozen other computational biologists contribute to that as well. So there's a sort of a close connection between the work of the biological network and that core team that assists with integration and benchmarking. So the components of this snapshot include, starting in the upper left, obviously you see who the coordinators are. They have to describe how the biological network was involved, how the landmark papers were incorporated, and how integration methods were benchmarked. They have to provide links to the code, the matrices on cell by gene, papers, and any other information there. And then on the right side, you'll see commentary from both the integration team, particularly Malte, and the coordinators themselves about what sort of the key takeaways are and some of the key considerations were for building this atlas. And so every network is asked to provide these snapshots, and they're actually very helpful for summarizing the network's approach, which again, you'll hear much more about from Ray in a minute. So then on the next slide, once the atlas is approved, then we work with the operators of the HCA data portal to get the atlas information up on the data portal. And for those of you who are frequent users of the data portal, you know that over the last 18 months or so, we've really started a transition to focusing on more and more on atlas content as opposed to the individual source data sets. Now, the data sets are still visible through the HCA Data Explorer, and that the button to the HCA Data Explorer is there at the top middle of the slide. But then down below, you see all the icons for the 18 HCA biological networks, and you can see the little blue dot there, the little blue button that indicates which of those networks have already released an atlas. If you were to click on the iNetworks icon here, it would take us to the next slide, which is the iNetworks homepage on the data portal. And here you see a general description of the network itself, contact information for the coordinators, and then at the bottom, you will see a list of all of the atlases that have been produced by that network. And so, of course, right now, this is a single atlas, which is the Retina Atlas. But as you will hear from Ray, the iNetwork has many plans for additional atlases covering the rest of the eye. And so those atlases would also appear here. And then if you were to click there on that single cell atlas of the human retina link, it would take you to the next slide, which is the actual atlas page on the data portal. And here, again, you see a general description of the atlas. There's actually a second tab, which is the list of all the source data sets and links out to where you can find that on cell by gene as well as the HCA data repository. On the right, you see the links to the code, publications, contact information, and so forth. And then down at the bottom, you see what we term component atlases or sub-atlases. And there's actually more than 2. And on the next slide, I'll show you that. But for now, I will show you that in addition to a general sort of listing of which sub-atlases make up the Retina V1 atlas, you can also see links out to cell by gene, where the integrated object is available, and you can actually download the atlas directly, or the component atlases directly from this webpage. And that's a collaboration that we have with Cell by Gene, and we're really pleased to work with them on that. So on the next slide, I will hand this over to Ray, but this is actually the full list of component atlases for the Human Retina Atlas V1. I'll ask Ray to sort of take you through this on this slide and the next slide. So Ray, thank you again for joining us, and please go ahead.
Yeah, thanks, John. So this is Chen. Just thank you for the opportunity to share our experience, of the process to get the atlas up and loaded, and then so this, as John said, once you click into the portal, and then we have actually subcomponent in addition to the atlas. We also have subcomponent because probably true for many tissues. There are cell types. We have more than 120 cell types, and then some of the cell class have very complex. For example, immigrant cells have 70 cell types. So when we try to identify the cell types, we need to have different resolution and help people to annotate and navigate. We actually pull out those subclasses and then set them into a separate atlas as a sub-atlas. So in addition, you will find at the bottom of the line, there's also a taxic data that is also get pulled out separately from the single the rest of atlas. Another nuances is the retinal atlas are composed both single nuclear RNA-seq and single cell RNA-seq. We also have a subset of this data. So you see the next slide. So I just want to quickly give a summary on what this current retinal cell atlas version 1 looks like. So it has 7 published and newly generated data set. The 7 landmark papers, as John mentioned, is important to be part of the atlas. And also there's actually currently have 3 million single nuclear seq and single cell RNA-seq combined, 110, 120 cell types in the human retina have been clustered and annotated right now. And we're also in this particular one, when we try to annotate, we also borrow the cross-species information, like the study in terms of cell type are majorly done in a mouse, and then we want to borrow just a lot of conservation between these two species. We borrow a lot of information from other species, including mouse, but also including non-human primate studies. So we have in companion, we have the single nuclear taxic data, and we identify more than 600K open chromatin regions across different cell types. And also another thing, because we have the RNA-seq and the taxic, so that allows us to try to fan mapping the eye-related GWAS site, and that's actually in the publication, and then hopefully it can be annotated properly here as well. So next slide. It's very critical to get the community involved. So they're involved in many different ways. The first way is that we want to basically include the key paper publication, publish the data set. So they contribute directly to the data and analysis. So the important things here, like a lot of paper when we publish, they have the cell by, they have cell by gene like count matrix, but they may not have the raw data. So we actually spend a lot of time and then to get also the approval, so we can get the raw data, so that can be integrated with the rest of the data consistently. Another thing actually very important, like at the tail end, is also it's very important to involve the expertise in the community to curate the cell cluster properties. I will touch that a little later. I think that's really a fruitful exercise, and then it's not only try to ensure the quality of the annotation, but also get, as a community, we actually discuss on what's the next version, and then also there's a lot of potentially a collaboration can come out of it. So by including the data published, they have each paper have their own annotation, and in fact in the retina there's a multiple set of way how to name cells. So it's important to record those information, and then by combine them everything together, we have the confusion matrix, so to speak, so that we can see what's current cluster are corresponding to what cluster in the previous paper. So that's really helpful for researchers to navigate the data. So next slide. So as to track the as John mentioned, in addition to the data portal, here the data also are deposited to the cell by gene collection, and then the website and database, and also on a CAP. So this is a tracker that we can easily look at the progress, how this data are ingested and then uploaded to different platform, and then you can see in the retina we are on HCA, of course, and also in this particular one, we're probably still working on get everything on cell by gene, and then also currently already on CAP, and then we're just starting that process for annotation. And then to the right is the status of approval, whether it's a version 1 atlas. You can see it's green in the retina, so meaning we are already approved. So the next. So it's a bit hard to see. So, and this is more detail. If you click in, this is more detail on all the data sets made the atlas, and then you can see their current status on where the data is at different platform. Go to next. So maybe right now I will spend the next few minutes to dive in a little bit on the atlas data itself, so you can see more clearly as example what the process. So this is the data component of the retina cell atlas version 1. So in blue are published data sets. So you can see there are like right now actually there's eight studies, and then there's also in the green are the ones that have been generated but has not been published. But it's also part of the atlas one. So totally we actually just upped the data to 3.4 million. And from 122 donors. Next. So we try to be, as a reference, we want to be as representative as possible. So we have balanced donor representation in terms of ancestry. So we have maybe slightly more than half are white, but also we have 40% are other ancestry group like Hispanic, Black, and then Asian. And then there's some unknown ones. We actually do computation and then assign them because unknown was initially from the publication. There's no record for it. So about 65% are male and 35% are female here. So at the bottom is showing the age distribution. We have from day 0 to all the way to 94-year-old, and try to cover the aging process. And of course, you can see the harder part is really the pediatric ones because it's much harder to get the tissue, and this skewed to the more elder population, but we tried our best to cover them all. Next. So this is like low resolution quick look on the atlas look like, and then each color represent the major class of the cells. And you can see, for example, amical cell in blue to your left, it is a lot of small clusters because they have most complex cell class, while some others are basically just one color because it's just one cell type there. So since we integrate all the data, so we take all the raw data from published data and also our own data, we run through the same QC pipeline and matrix, and then we did benchmark on how to integrate, harmonize it. We used, in this particular case, we used single cell SCVI and to perform the remove battery and integration, and seems to perform quite well. And then all the pipeline, the process are on the GitHub that people can use to reproduce the process. Next. So in addition to the single cell, single nuclear RNA-seq, as I mentioned, we also have single nuclear attack-seq to looking for open chromatin state. And this is and then the resolution of attack-seq is much lower than the RNA-seq. First, they have less number of nuclear profiles, but mainly really just the technology itself. And then, as you can see, so to improve the resolution, what we did is we did co-embedding between single nuclear attack-seq, a single nuclear and single cell RNA-seq, and to help to annotate and improve the resolution of attack-seq. You can see they match very nicely after integration. Next. So this is a quick look at the single nuclear attack-seq peaks, and then we totally about 600,000 peaks identified, and then this data have about 500,000 single nuclear attack-seq profiled. Next. So the data has been uploaded in addition to the, it can be accessed through the, Atlas data portal, but also it's on different platform. So it's on cell by gene, but also because we have attack-seq data, and then we also upload into the UCSC cell browser and the attack-seq And then also you can visualize the ataxic data in browse. So those are all the cells, just so that, depends on and since different portal have different, how to say, strengths, and then so for people looking at different aspect of the data, they can choose which portal they want to use. And all those links are on the Atlas data portal, so you can simply click and then you click to those database. Okay, next. So I think the last day, once we have the initial annotation, and then our postdoc and the researcher, by combining the information from previous annotation and also marker genes, we perform the initial annotation, and then at that point we load into the CAP system, cell annotation platform system, that developed by Chloe's group, and then we work very closely, I will say, with Yvonne and Mary, and they're tremendously help us to put the data on the CAP system. And this nice interactive system is actually a very ideal way to help us to get all the community input, and people form. We form, basically what we do is that we reach out to the community, and then thinking about, depends on the cell class, some people and basically bring in the expert who actually in that particular cell class, they have done not only probably molecular level, genetic level, but also physiology and morphology and function level, and then to get together to basically start annotate and curate the annotation we have initially. So we currently, for the eye, there's a 5 working group have been formed, depends on which part of the eye, and then even in the retina we have 2 subgroups. One is for amacrine cells, one is for RGC cells, because nobody have expertise of every cell type or classes. Many of them has never been seen before, so that's actually really require a collective wisdom. So as Shyam mentioned, in addition to retina, we also have other parts of the eye coming through. For example, cornea surface, eye surface, triplex meshwork, and those form a different subgroup. And totally, we have for the retina, we have 40 researchers actively working together. Have a Zoom meeting, and then also all log in to have this interactive mode to annotate and to vote, and then to discuss the nomenclature. I think this is actually a very good exercise because, as I mentioned, the retina, there's like at least three system to a normal to name the cell. I think this is actually an opportunity for the community to come together, that we can come up a good nomenclature system that we can hopefully use going forward. So it's just much easier for people to exchange information going forward that way. Once we have a consistent nomenclature. So I think that's it from me. And then thank you for listening.
Thank you very much, Ray. We'll have a few minutes now for questions, and I would encourage people to put questions into the Q&A, and then we can relay them to Ray. We're also joined by Chloe Villani, who is the lead PI on the cell annotation platform, and so she can answer questions about that. She also has roles, excuse me, in the HCA Data Ecosystem Oversight Group as well, and so she can also address other questions. I want to actually thank Ray, not just for his presentation, but also because the Eye Network has really been a wonderful partner in not just helping to get all their information onto the platforms that you've heard about, but also helping us sort of think through these processes about posting data and metadata, and ensuring that there's alignment between the HCA Data Repository, the cell annotation platform, and Cell by Gene. And so I want to thank him for that. I also want to thank our partners who operate those platforms, both the CAP team and Cell by Gene, but also the organizations that operate the data repository and data portal for HCA, which includes EBI, the Broad Institute, the University of California, Santa Cruz, and the firm of Clever Canary that's responsible for the design and build of the HCA data portal. So this was a really wonderful presentation, Ray, and I think as we're waiting for some questions to come in, I actually would like to ask you to maybe expand a bit on the other working groups. What are the plans for the other elements of the eye, and how do you think about going beyond V1 and actually bringing those different atlases together for cross-tissue or at least intra-eye annotation.
Yes, thanks, John, again for the invitation. So yeah, so in the eye, I was thinking, eye is like a mini body. So the we're having a focus on the retina because this is the major target for irreversible blindness. But of course, there are all parts of eye is necessary for the eye to function. And then we have generated data for the entire eye. And then because the the community actually who work on the anterior segment, for example, the eye surface cornea, are actually not expert on the retina, vice versa. So we organize based on their expertise into different working group, and we have basically the way we do it is we have the quarterly eye network meeting, and then we invite people who are expert and in different area, and then we have to ask them to volunteer on which part you'll be most interested, and then you have expert did on, and then so they actually sign them up on different parts of the working group, and then for each one we now already finished generating all the data, and then so those data are in the process to upload to a CAP system and to the cell by gene, and then now they start to basically have regular meeting to start annotate and curate the annotation. So they're a little bit behind the retina, but in fact, they probably finish in terms of the cell curation sooner than retina, because retina is the most complex ones. The other ones are slightly more straightforward. So going beyond, we are interested in one atlas, for the entire eye, but I see there's a lot of challenge. For example, fibroblast cells exist in all tissues, and then sometimes how are you going to integrate them together? Those types of things we are still try to test on, but it could be quite challenging for some of the cell type. Some cell type are very distinct, that's very easy, but there's some cell types are, and also residential, for example, macrophages, and those cells are we need interaction with other networks, for example, for immune, how can we deal with this, that sort of thing. Yeah.
Great. We have a few really good questions in the chat. I'll just take them in order. The first is a general question, which I can provide an answer to, but invite Chloe and Ray to chime in. The question is from Vittoria Bacci, who asks, if you have an integrated atlas that could fit well with the HCA, who should we reach out to for integration into the HCA? Excuse me. I would say the starting point for this is really the relevant biological network. I would encourage you to reach out to the coordinators of the biological network. There's contact information both on the HCA data portal page for that network, but also on the main HCA website. If you go to the section for biological networks, you can find the contact information and you can get in touch. That's really, I think, the best way. I think if you feel like your atlas is not covered by one of the 18 networks, hopefully that's not the case, but if it is the case, then I would suggest maybe as a starting point, as I always suggest on everything, reaching out to the executive office as a first point of contact, because then we can put you in touch with the most relevant people to answer the question.
And the contact information for the executive office is also on the HCA webpage. And perhaps, John, to just add a few words to your answer, I would say we're very eager to hear from all of you. If you have additional data sets that are about to be published or just published, it's not because we're releasing version 1 that we are done. We're far from being done. It's just the beginning of the journey. Aviv mentioned that we're very cell-centric. We're going to talk about spatial a little later on in the town hall, but that doesn't mean we stop being being cell-centric. We still are eager to continue integrating single-cell suspension to also help increase the sampling of every tissues we're studying, because of course we were not that thorough. I'm sure that Ray wants even more data if they're available, as we work towards our version 2, which will include more cells, eventually more modalities and space. So again, just want to highlight, it is not because you're hearing about version 1 that we are done, and that we're done in terms of the single cell RNA-seq suspension cells. There's going to be more opportunities as we continue versioning in the years to come and updating our nomenclature. Ray, anything else you'd like to add?
Yeah, I mean, absolutely. We're having in contact recently on a potential other data set coming up. Although we have like 3 million, single nucleases for the retina, which is very small tissue, sounds a lot, but actually we're still missing coverage for some extremely rare cell types like RGCs. And so we'd love to have additional data. We have, as Chloe said, we're actually in the process of get into the version 2 by integrating spatial data and then also extend to the disease status. So there's opportunity for us to include that into the next version. So we're actually working on that. Right?
Great. The next question is from Sirsha Sankara, who asked a question on a topic that we talk about a lot in HCA, which has to do with quality control of data. So the question is, for data QC pre-processing, would you consider a value add in implementing a data-driven QC approach as opposed to a data agnostic approach, as in running through a standard QC pipeline? And then the point is made that QC criteria can be a bit more rigorous, which is probably a nice way of saying variable. So let me ask, I think it's really great to hear from both Ray and Chloe on this point about data QC and productive approaches.
Well, maybe I'll go first very briefly. I want to clarify that the answer is gonna be specific to or tailored to every biological network and every atlas. Everybody is approaching this a little differently. Everyone is encouraged to use a different QC approach to ensure that we maximize the quality and reproducibility of the atlas. And we encourage everybody to try, benchmark, compare different approach for QCing their data. I will let Ray address the approach for retina. I can briefly speak for the immune bionetwork, for example, since I'm involved with the immune integration, where we have used both some methods that were more automated combined with some methods that were very manual, to kind of accelerate the process of identifying cells of poor quality. So no, I just speaking in terms of my own terms, we don't just go blindly with the automated QC output. We use it to kind of help us hone in into where we should go and manually look at some potentially poor quality cells. But the bigger picture answer to your question is that there's no one size fits all solution. And this needs to be tailored to every effort, every biological network, and every type of data modalities that are being looked at. Ray, would you like to
I
address
think
this?
yes, I have very similar experience where we run through the standard pipeline, example, mitochondrial content and then the cell count, but I think when you move downstream, then you will see potential doublet removal is not like complete by the software, and there you also will find samples, for example, at sample level, the multiple level. At sample level, some cell clusters are entirely coming from one particular individual, maybe something. Our data is from postmortem samples. They have, the donor has experienced their unique experience basically. So those things we flagged it, and then some of them we decide to remove it. But we do keep all the data still in there, and then just have the annotation and also have the code to say what we exactly have done, so you can reproduce it. But I think the QC, yeah, I mean, as Chloe said, it's a combination of automatic automation pipeline but also manual looking at it and make some decision.
And perhaps just to add a few points that ultimately we are doing data-driven QC and much more in terms of ensuring that as we build these gold standard reference atlas, we're maximizing high quality data set, because it's very key in driving the development of consensus cell annotation as a next step.
Great. The next question is a general one, and I think it's really great, actually, Chloe, that you're here representing CAP, because this question is, could you please describe your format for community engagement around cell annotation? A really critical question. I'll ask Ray to answer this first, and then Chloe, you may have additional points.
Yeah, I will, as I mentioned, the CAP is actually a natural platform, very ideal for the community engagement, because everybody, get a login, and then they basically, and they are assigned to a particular working group, and then they can basically record all their comments. This platform that allow you to when you're doing annotation, you can disagree or agree with previous annotation, that also put your rationale behind it, and those are all recorded. So it's really like a forum, so that you don't need to be physically or on Zoom together. You can work independently. And then what we do is we have everybody log in their information, comments, and then we have a Zoom session or in-person session to discuss those. And and highlight ones that there's more conflict, and then really just quickly skip ones that actually consensus. So I think this very natural, very nice platform, interactive ones. And then the key thing is all the comments are recorded there, so you can always look at all the history and how this annotation evolved. So we consider this annotation is a living document. It's not like snapshot. So it's really continue to evolve. So we will gain more and more knowledge down the road, and that allow us to better and better curate the cell clusters and also make decision on merging and splitting and so on and so forth.
Thank you, Ray, and I'm really thrilled to hear that you like the features presented on the cell annotation platform. We're always eager to hear feedback on how we can improve and empower the community. To take a step back and to kind of try to frame my answer, I'll start by saying that something we often say on our end with the CAP team is that cell annotation can be seen as an opinion among biologists, and to be able to structure a conversation and compare notes, what we try is defining in a more objective way cellular identity in the single RNA-seq space using 12 fields of metadata for cell annotation that are well documented on CAP. And I'll go through them in a second, but we spend time trying to train the community on how to use it. So if you're interested in using CAP and haven't been in touch with us yet, do reach out. Also do reach out through the bio network that you mostly fit with, because most of them are already in touch with CAP. So these 12 annotation metadata field, to kind of give you a context, we're talking here about like what name do you give them, what marker genes did you use, were they data driven, are they legacy markers tied to them, are they non-cell ontology term, what is the closest ontology term if it doesn't exist. So it's systematic. And did you annotate manually? Did you annotate using an algorithm? And this is all compiled with very clear definitions of terms for every field. And that's tied to a user-friendly interface, so you don't need to know how to code, so you can engage with an embedding and understand the cellular identity assigned by the integration team and the Atlas assembly team. And with that, there are different features. As members of the community, you can go in, you need to sign in on CAP. It's free, it's open access, and we do record everything publicly, so you should be aware of this, because it's meant to be a transparent and reproducible process. So you can go and engage, sign up with your email, and then you can go and say, for example, I'd like to help with the retina annotation with this specific cell subset. I have experience in it. So you could go in, even if you're not involved in the Atlas assembly, we actually want the community, we want to hear from all of you. So you can actually come in and say, oh, let me see, they've defined 12 subset here. Do I agree? And they're very easy buttons. You can say thumbs up, I agree. And if you disagree, you can disagree, but you must tell us why. And there's a framework there. And you can say, I disagree with that this is a unique entity. We think it's two entities. Split your clusters, and you tell us how to split. Or you say, actually, these three subsets, I think it's one, and you tell us merge. Or you can say, I agree with the molecular definition, but I disagree of the markers you use. And you can tell us how you would change it. And as Ray mentioned, all the comments are compiled. And it will help us as a community define which cells do we achieve broad consensus and which cells do we disagree, and where we should spend more time as a community to work on either sharing more data or have live meetings to kind of work out these discrepancies. And so it is meant to be a user-friendly interface to support the live atlas, as Ray mentioned, because as we get more data with different modalities, of course, cellular identity will evolve and change over time. So we'll do timestamp and say at this moment in time, this is what we have, and this is the type of consensus with reach. And it also enables everybody around the world to engage with it. You don't need to make it up to a meeting at a specific date. Everybody's invited to join, and that's one of the purpose of this annotation platform, is to kind of make it equitable and accessible. And we invite you all to reach out. If you don't know how to navigate it, you can reach out to CAP. And as John mentioned, you can also just start by reaching out with the executive office if you're not sure where to start, and they can orient you.
Great, thank you. We have time for one and maybe two more questions before we need to move to the next segment of the program. I do want to mention, I just encourage everybody to keep on asking questions in the Q&A, and the speakers, please feel free to answer those questions in writing if we are not able to get to them live. But these are all really excellent questions. The next question comes from Mike Benben, who asks, do you have any plans for incorporating electrophysiology data into the Retina Atlas?
Yeah, that's definitely a next step. That's a holy grail in a sense, right? There's two community, one more on molecular biology, one genetics, and the other on morphology and physiology. We're actually definitely thinking about that. I know there's a recording, technology like MEA get much better, so people can record, the physiology much better than before. And also there's EM data that looking for connectome and the morphology. So yeah, we're actually actively working with people in our community. For example, EJ from Stanford is on our panel and try to give the opinion on how to better annotate the cell type, and then also how can we have this interaction, collaboration, potentially next direction, how to merge these two. I think that's critical. Yeah.
And I'm sure there are parallel examples in the other networks as well. I'll take one more question and then we'll move on. But again, we can ask and answer questions in the Q&A. So the next question is from Dua Mugahid, who asks a really actually pertinent question for the retina, which is, are there plans or interest in incorporating data from non-human primates or even mice? Which is a great question because Ray and his colleagues have also in parallel produced a mouse retina atlas. So Ray, do you want to talk about your thinking on this topic?
Yeah, I think that thanks for the question. I think cross species serve many purpose. One is help to annotate human cell types because we know way more things about mouse than human, fortunately or unfortunately. So but also since our most study are actually conduct on non-human primate or actually mouse, and then how you translate the information that you learn from mouse to human, for example, is critical, right? In terms of understanding human biology and human disease. So yeah, so Josh Chen is also a close collaborator and also on the panel. They have generated recently, as you probably know that a single cell profile for many species across and then perform the cross-species comparison. This is very active research field, and then we definitely are very engaged in that aspect.
Great, thank you. I think with that, we'll move to the next segment of the program, which will talk about the future priorities for HCA. We've already, of course, been introduced to Chloe Villani, but I'll also ask Shyam Prabhakar to join us. Both Shyam and Chloe have many roles within HCA, but I will start with that they are the co-chairs of the Data Ecosystem Oversight Group. Chloe, in addition to what you've already heard, is also a coordinator of the Immune Biological Network. Shyam is a coordinator of the Genetic Diversity Biological Network, and it's also co-chairing the Diversity Task Force under the Equity Working Group within HCA. So there are a lot of different working groups and networks that are all collaborating on some of the questions that you are about to hear about. They'll recap some thoughts about they're coming from numerous groups within HCA about future priorities and where HCA is heading beyond V1. So thank you, Chloe and Shyam, for joining us, and I will turn it over to you.
Thanks very much, John. All right. A fun part of this presentation, you've heard the amazing tales from Sarah and Aviv about the HCA publication package, which was a great success, really a landmark achievement for this consortium. You heard also from John and I think from Sarah about how the V1 atlases are coming together, and Ray presented one of them, the I Atlas. So in a sense, 2025 is a transition point, you can say, or a cusp in HCA's evolution. Whenever you reach or approach a finishing line in science, you look back and you say, okay, great, we did a lot, we achieved a lot because we had clear goals and we focused on them, and that's how we got them done. But then when you reach the finishing line, you look back and say, okay, there's much more that needs to be done, right? For the next phase, there's many more things you can add to our goals to really understand the human body and build a representative atlas. So that's the motivation really for our atlas expansion strategy in the HCA, as you see here. Technology has advanced. So where single cell was the cutting edge technology when HCA started, now spatial has complemented that. So definitely a big part of HCA's expansion will be using spatial technologies to build a 3D atlas, going from single cell to 3D, right? So just to recap again, our successes so far have been largely because we had a clear plan about using mostly single-cell assays in individual organ-specific bionetworks to build primarily healthy atlases. Okay, so the key pillars of our expansion strategy will be to go beyond all of those aspects, going from single-cell to spatial, and then going from healthy atlases to applications, disease applications of healthy atlases, which will be really essential, as I'll mention, to our goal of being equitable as well. And then also we'll go from individual organ-specific bionetworks to cross-tissue reference atlases. For example, in the Q&A just now, somebody asked about, can you query B cells across all organs? That's precisely the goal of this kind of cross-tissue reference atlas effort in the HCA. So that'll be the next phase. And of course, underpinning all of this is our commitment to diversity and equity in the HCA. And so all of these initiatives will have a diversity commitment, as I'll describe in a few minutes. Next slide, please. Okay, so starting from equity and diversity, right? The HCA Diversity Task Force met over almost a year, I think, and deliberated, reached out to a lot of people. And so I'll briefly summarize the work that we did and finally the report we generated for HCA. Starting with the motivation, okay? So above all, equity is a fundamental value for HCA. It starts there, but then there's also benefit. There's also clinical benefit, right? That why are we looking at diversity from a benefit angle? It's because our bodies are different, right? We're not all the same. So our molecular, cellular, and tissue traits are variable across humans. So, and that's certainly important for identifying healthy baselines for diagnostics, but also disease risk, a likelihood of getting diseases, disease physiology, drug response when we treat it is again variable across humans, right? And so that's the whole point of precision medicine. One size doesn't fit all. We have to accommodate, understand, and then respond to all this human variability, right? So again, that really means it's incumbent upon us to build an atlas of not just one human, but of all humans. So that's a benefit angle. And then there's a science angle too, which is we know so little about human diversity at high resolution, right? And that's where Human Cell Atlas can make a difference. We have a global reach. We have almost 4,000 members. We're in over 100 countries. We are the best positioned global scientific consortium to address this exciting new scientific frontier, right? But of course, we need concerted efforts to make a difference, to move the needle. We've done well so far, but definitely far from our goal, right, in the diversity realm. So going from intentions to strategy is what the Diversity Task Force was tasked with. Next slide. Now, before we get to the actual research, the bulk of the Diversity Task Force recommendations were about how you do the research, okay? But to be able to do that research in a representative way across the globe and to get participation from researchers all over the globe, there are definitely challenges we need to address, okay, which we've faced throughout. One is simply getting funding to the respective teams so that funding is not concentrated in a small number of locations, and then training and capacity building all across the world, which includes travel, of course. Capacity building also includes infrastructure, and that means not just wet lab infrastructure, but also IT hardware and IT systems, trained IT personnel. And then community engagement in various HCA locations so that the local communities understand the benefits and, of course, the risks of this research and we understand what they are looking for, and then you have a motivated set of participants and partners for your research, right? And finally, from representation participation to benefit, as I'll talk about briefly, disease applications of our healthy atlases will be essential to motivating people all over the world to participate in the HCA. Next slide. All right, so coming to the research side of our recommendations, human diversity means many things to many people. We decided to focus on these specific aspects of human diversity. This is genetics, genetic ancestry, self-identified ethnicity, geographical location, sex, gender identity, age, and environment. You can say, these are the broad axes or dimensions of human diversity we want to understand in the HCA, right? And to do that, of course, we made some recommendations about ethically and scientifically appropriate processes, though this mainly took the form of referring readers. You should hopefully see our recommendations soon, once they've been ratified. Directing people to the existing bodies that have already put out guidelines for this kind of research, okay? And then going to something very specific, which is to be able to scientifically study diversity, you need to be able to understand all the covariates, right? For each sample, each donor in the HCA, you will ideally be able to know all of these attributes of the donors, their genetics, ancestry, ethnicity, geography, sex, etc., right? And that's where metadata comes into play. We need a harmonized metadata spec across the whole HCA, across all the bionetworks, so that we can do similar analyses in a harmonized way, right? So we came out with a very specific metadata, tier 2 metadata specification, which we hope you will all adopt. Tier 2 means it'll be access controls, so that'll protect privacy. But researchers who have access will be able to use the metadata to understand the molecular and cellular correlates of human diversity. And then tracking metrics, we can use what people submit, with the metadata that people submit, to track how well we're doing. And that's really important, right? We can talk about diversity as a goal, but we want to also be able to quantify how much progress we made at any given moment. And lastly, we want to set clear targets. And this is where we turn in a most practical way, right? We turn our intentions into clear implementation, right? And here you can see a sketch in this figure of our quantitative goals. We've set 5-year goals, we've set 10-year goals. The most specific quantitative goals are for our geographical diversity, because we believe that with geographical diversity, we'll get ancestral diversity, we'll get ethnic diversity, and so on. And so this takes the form of the number of countries from which we'll get data for a certain number of bionetworks, blood being the easiest. Blood already has the most diverse data sets. What we want to expand to, in 5 years, we want blood data from half the countries in the HCA, and skin and nasal swab from, say, 30 countries in 5 years. And then in 10 years, we want an even larger number of bionetworks, which would, those are the more challenging tissues to get to 50 countries. So ambitious goals, but we really believe that this is necessary for all the reasons I mentioned on the previous slide. Next slide, please. In terms of the metadata, these are the fields that we hope you will all collect in your research. This is a very high-level outline, but you can see that the ones in black font are the metadata fields that are priority fields. The ones in blue font are the recommended fields, which will also really greatly advance our understanding of human diversity. But we understand that these might be harder to collect, right? And so we put them one tier down. But of course, and actually even the ones in black font, they could be sensitive That's really between you and your IRB and your community to decide what you can ethically disclose and what you can't. Next slide. So coming to expanding representation, expanding participation, achieving benefit, and so on, one thing we realize in the diversity task force as we talk to people in the community is that many countries really need a disease angle in their research in order to receive local funding and to show benefit for their local communities. So really they wanted that as a motivation to join and to actively participate in HCA. By that, I mean disease applications of the Healthy Atlas, right? And of course, as I mentioned, we need disease applications studied in diverse locations all over the world to build a scientific basis for precision medicine, right? So that's the motivation. And certainly the HCA recognizes that. So this is remarkable, really, that the HCA listened to the feedback from the diversity task force and recognizes that, okay, disease application should come in phase 2. We already have, of course, it's not to say we didn't have it. We already had major advances for COVID, where HCA contributed. HCA members have worked on Alzheimer's, many other diseases. The goal is to expand that, right? And so how do you expand that? How do you pick a small number of diseases that we can work on and prioritize initially for HCA focus? Because obviously you cannot tackle all diseases. So these are the criteria over here. You want to look at disease burden globally and locally, look at rare diseases, look at neglected diseases, get feedback from all the regional networks, understand the capabilities of HCA members, get feedback from the buyer networks as well for specific organ diseases related to specific organs. And also we want to keep an eye on what other consortia are doing so that we are complementary with them rather than replicating what they do, right? So that's the broad vision and motivation for disease applications in the HCA. What's the next step? We really want to get your input on which diseases to prioritize, and that's the goal of the survey. So stay tuned. So what tissue samples you've been banking that you have access to that you could start on quickly? Which diseases are your priorities? What types of assays you would like to do? And then what are the flavors of disease you're interested in? And then based on all this feedback from the HCA regional networks, from Asia, Africa, Middle East, Latin America, everywhere, then we can build up some kind of coherent plan, right? So that then we can, based on feedback, we can work with buyer networks. If there is an organ-specific buyer network or organ-specific disease, we'll work with the respective buyer network to organize that effort. Or we could classify the disease as a systemic disease, in which case it would be across buyer networks. And then we'll have our prioritized diseases that hopefully will be, all of them will have a diversity angle. All of them will be worked on by multiple teams across the globe, right? So that's the broad vision for diversity and disease in phase 2 of HCA expansion. And we hope you'll all be part of this conversation in defining, in expanding diversity and defining our disease priorities. So step 1, please complete the survey, which you'll receive in a couple of weeks. Thank you.
Thank you, Shyam. And so I hope you heard him loud and clear. You all have a chance to help shape the next chapter of HCA as we embrace more broadly studying disease. Next slide, please, which is a unique opportunity to show a direct application of these healthy atlas to address these global disease burden, but of course, also achieve our overall goal of being an equitable endeavor. Next slide. And you've seen this first slide when Aviv was presenting, but just to kind of bring it back into context through what Shyam just presented you, Of course, version and Ray just presenting you. Of course, version 1 of our healthy reference has been very cell-centric, like Aviv said, with a lot of single RNA sequencing. As I mentioned during the first Q&A, it is not the end of the journey, we are still in the beginning of this journey of adding additional data sets in terms of additional data sets of the same type, so we can have better sampling of a tissue if they're available, adding different modalities, including multiomics and spatial, and increasing the diversity of the cohort that we're analyzing. And as we continue doing this, which is the main priorities for HTA, we heard the community loud and clear. We're going to start also applying some of the atlas through the lens of studying some disease. And again, you're invited to contribute in shaping that roadmap of which disease will be prioritized in terms of these global and local disease burden, those that are very important to you in terms of the different regional network, so that we can start applying some of our atlas to disease context. And as Aviv mentioned, there's quite a lot of potential benefits, of course, with our overarching goal of being more equitable. We also realize that many of our community have been biobanking a lot of tissue blocks over the years that are readily available, FFPE blocks. And so one way of increasing and doing capacity building and increasing the involvement of the broader community is actually to embrace studying some of these diseases through the lens of our spatial program, which I'm going to talk about in a few minutes. And overall, we're hoping that through the lens of these new endeavors, we'll be able to benefiting advancing the knowledge about high burden and neglected disease, and also leveraging the fact that we are building atlases across organ system and are working towards integrating our data across organ system to have an overall view of the system, which would uniquely position us to start tackling multi-system disease. Next slide, please. And in terms of what we're looking into doing for applications, you can imagine that incorporating healthy atlas with disease data, we can start mapping disease mechanisms through which may help us hone in on some potential diagnostic and therapeutic targets. From a biologic perspective, it will give us an opportunity to define the cellular context of relevant genetic association studies to different diseases. So what is the relevant cellular context of all these susceptible genes we've been mapping over the years? And beyond defining therapeutic targets, actually trying to move towards helping develop the next generation of regenerative medicines and therapy. Next slide, please. And so, as I mentioned, one way of approaching the next phase of HE, both through the lens of disease, but also starting to build our atlas an additional dimension, continuing some of the cell-centric approach, but actually moving more towards space, as Aviv mentioned, is really to kind of embrace these spatial technologies. And an image here is worth 1,000 genes, it's worth 1,000 words. And so the time is right. The technologies have matured. And next slide, please. And in the paper bundle that was released, there was already the first generation of spatial atlas, which are growing rapidly. They were data released from the GI tract, the thymus, the placenta, the prenatal skin, skeletal development, embryonic limbs, endometrial, and the COVID-19 lungs. Next slide, please. Along with some of the early computational methods development being released, there's a lot of work to be done in that space, but it's exciting to see several papers already coming up, whether it's pipeline to integrate single-cell and spatial transcriptomics data, or effort in trying to define landmark detection methods using deep learning strategies. Next slide, please. And so there's a lot of work to be done to empower the entire community. One of the things we want to do is try to define what are the major roadblocks right now in the broader communities to embrace these technologies at scale. And so what we're currently doing is assembling task force across these 4 tracks that I will briefly describe. And the mandate of every task force is to precisely define these roadblocks and to prioritize them so that we can organize activities around them to alleviate these roadblocks. And when I say activities around them, it can be part of meetings, it can be fundraising projects. So I'll give you some examples. The first track is about optimizing and comparing technology platforms for enabling high quality data productions. there's an increasing growing landscape of different type of spatial methods of microscopy, whether you're in the RNA, protein, or other modality space. There's a need to map the landscape of these technologies. And then it's not about finding a winner among all of these. It's about defining what technology is good for which questions and for which tissues. So we're actually working with every biological network to figure out what are their requirements. And this task force ties to the Standard and Technology Working Group. The second track is tied to algorithm and analytical pipeline development, which is tied to our analysis working group. And there's a lot of work to be done in this space. And this is not only about developing necessarily a new algorithm, but also trying to make recommendations and defining what are the roadblocks. I'll give you an example just to contextualize this. There's a lot of work being done in terms of developing optimal cell segmentation strategy. But there hasn't been effort in comparing, contrasting at scale these different type of strategies. So that could be an example of a roadblock if we decide that this is a roadblock that we could tackle. The third track is related to generating data, both in health and disease, and actually working with all these other tracks here and generating the right type of data and then developing the right analytical framework to integrate spatial omics data with our single cell suspension. So that's work to be done very closely with track 2. I'm going to mention this on the next slide, but we want to work hard with different for-profit partners to develop spatial panels that the entire community could leverage. We also want to work with different pathologists so we can think about integrating spatial omics with histology-based diagnostics. And so this track is tied to every biological network representative and to the data ecosystem oversight groups. And finally, the fourth track is tied to portals and data visualization. As we want, a bit like CAP enables you to work with annotation, it's important that the entire community can navigate and query the spatial data. And so that's about working towards analyzing spatial omics with single cell data and other modalities, with different interacting visualization that are intuitive and with equitable access. And that track will also report to the data ecosystem oversight group. Similarly to the disease survey, there's going to be a parallel survey. They're both going to be very short. That we're going to ask you to answer. We want to understand who is doing spatial, who has tissue already banked, but has not started doing spatial, but would like to do spatial. We're trying to organize the community to try to see where are the different efforts and how we can prioritize different fundraising opportunities. And so you have an opportunity to be part of this planning step. You're gonna get invitation to different socialization activities. We invite you to join and be part of that conversation. And we hope to have a full report to present to you by the general meeting in Singapore in June, this coming June. And next slide, please. Other activities includes developing new standards for spatial data, metadata, and file format. This is part of the analysis track. So that we can empower the community to start generating data in a harmonized way and working specifically with every single 18 bio network so we can leverage the version 1 atlas they've been creating to derive uniform panels that the community could use to do spatial transcriptomics at scale. And so please be responsive to the survey that will be circulating in the coming weeks. And with that, I think we can take questions. Next slide, please.
Great. Well, thank you to both Shyam and Chloe. That was a really great overview of a lot of the discussions that have been underway within HCA. And I really want to say that these the plans and the ideas that you've really just heard about have not emerged out of a vacuum. They really have emerged from discussions such as this one and the Q&A that we're about to have, as well as the many discussions that we've been having at both the general meeting and other regional meetings of HCA. And that includes last year's general meeting in Milan, as well as the HCA Asia meeting in Hong Kong, both of which had somewhere between 250 and 300 scientists in person and many more online. And so towards the end, you'll hear more information about the next general meeting in Singapore, and we certainly hope you'll join us and participate in some of these developmental discussions. So we now have 10 or 15 minutes for Q&A from the audience. All of the questions have been really terrific so far, and I would just again remind you to put your questions in the Q&A, and we're being joined by all of our speakers, and so as we go through these questions, I will invite the speakers to weigh in. we'll start with the first question that was submitted in this area. Do you plan to mine existing public data for diseases with unmet clinical need and use it in HCA?
Again,
Certainly, and this connects to, oh, sorry, maybe I can take that. This connects to one of the later questions as well of which we should maybe address simultaneously, which is, there are many diseases, and which ones are you going to tackle, all of them or just a small number? And again, I should say, we're going to prioritize and tackle some very specific disease where we feel that there's a strong motivation and strong capability. And for those diseases, definitely we'll be mining existing public data as well as generating new data. But of course, as we've always done in the HCA bionetworks, for example, the lung bionetwork is an excellent example of incorporating existing published data sets and generating new data, and then integrating them together in a single bioinformatic framework so that you can build a representative atlas. The immune cell atlas is doing the same thing. So even the healthy atlases were built that way. And certainly for disease, we will mine existing data sets.
Thank you, Aviv.
I would just add from, discovering disease mechanisms is one part, and having medicines is a whole other journey, and it's very hard. One of the benefits that we have from something like the Human Cell Atlas, including from disease data, is that often by analyzing disease tissue and seeing the states of the cells, you could, not often, but sometimes you can realize that in fact that disease could also be tackled by existing medicines. This is known as repurposing sometimes and line extensions at other times, and it is a very meaningful way in which actually medicines reach their full impact. One of the challenges that often exists with diseases that we know less about is that we don't even realize that they could actually be benefited by something that already exists. So I actually would hold non-trivial hope that by getting this higher resolution understanding, we would have more and more opportunities for that. And that would be almost like a shortcut, not just to know something about the disease, but to be able to do something about it.
Terrific. And this really does relate to the next couple of questions. We've already discussed, I think, the challenges around heterogeneous diseases. There's also a plea in the Q&A to study TB, both because of the high global burden, but also because the HCA has a lot of experience actually with, particularly out of the COVID pandemic, in studying lung disease and also more systemic.
Can I just comment on that one really quickly? In fact, TB has been and is very actively studied by some of the HCA leaders, and I would especially give a shout out to Alex Shalek, who has done a lot of work in this area and is one of our co-chairs of the Equity Working Group. So of course, a lot more to be done, and the Gates Foundation has actually supported that work for years now, and there's you could think about it like a smaller network of people with a lot of commitment to that.
and perhaps to just add a few words to what Aviv mentioned, when we talk about these disease endeavor, it doesn't necessarily mean just new data generation. To the point you just made, there's a wealth of data already available that will prioritize on integrating and contextualizing, which helps define where there are gaps in terms of additional data type that are needed. And so absolutely, but we invite you to fill out that survey so that your favorite disease can rise to the top. That could include TB, but of course there's if data are there, we'll be considering integrating.
And to the questioner's point, I also want to point out that both Alex and others within HCA studying TB are actually working directly with both clinical partners and biobanks in the regions that were mentioned as well. So we have a presence in all of these countries and are actively working with them. The next question asks how the spatial technologies are going to be chosen for the spatial branch of HCA, and what features are really key for our consideration.
Yeah, I'm happy to I've already answered one question in the Q&A in writing, mentioning that we have a Standards and Technology Working Group, which is co-chaired by Holger Hein and Arit Rosenblatt-Rosen, and that provides a forum for discussion of technologies, comparing them, carrying out benchmarks, and we encourage anybody in the community interested in those questions to join the monthly meetings of the Standards and Technology Working Group. Beyond that, it's also worth saying that, depending on the tissue, depending on the preservation technology, depending on the context and focus, there may be different technologies. And so there are various pros and cons, and the obvious things are obviously field of view, resolution, plexing, basically modality, multimodal spatial technologies are coming out now and so on. So these are all different perspectives that we look at in the Standards and Technology Working Group. And of course, not only the measurement technologies, but also all the upstream questions about preservation, sectioning, and also the downstream questions then on data analysis, where we also have in the HCA community, of course, the Analysis Working Group that also addresses the downstream data analysis questions. And I'll hand over to Chloe, who has her hand up to help me here.
No, this was a great answer, and I just want to add that is actually why this is an excellent question you're asking, and it's actually why we set up these task force specifically focused on spatial, and there's a specific track that is focused on technology, so we can answer these specific questions. We don't believe that there's a necessarily like a one-size-fits-all solution, but there's a need to understand what type of technology, and what type of modalities are needed for which questions. There's also a need to align on some of these measurements and some of these panels, because it will be very challenging to build an atlas in 2, 3, and 4Ds if there's not some forms of alignment. So that's both in terms of the technology type that will need to be assessed with different pilots, and also in thinking about how we develop these panels. Some can be unbiased, but also they need some levels of alignment. And we're so uniquely positioned here at with the UMass Atlas, with all of our version 1 work that is going on, where the team has been really hard at work, the integration team, and the Analysis Working Group and the Bio Network, in creating these high quality references that we can all leverage together to derive the most optimal panels to pursue some of these spatial endeavors. So we can balance unbiased and panels, which tackles one of the questions later on about how to make this more cost effective. And I'll invite Aviv to add to this answer.
I will say we actually are learning a lot also from experiences that have already happened in HCA. So I will especially highlight the work from the BRAIN Initiative, as part of which of course for us is the Brain Bio Network. There, in fact, multiple spatial technologies have been deployed, and initially most intensively, not in the human brain, but in the mouse brain, in the incarnation of the BRAIN Initiative known as BICCN. There's multiple things we learned from that, including the fact that it is advisable not to have a single technique because the techniques do complement each other, but also very importantly, that once we have a certain amount of data from multiple techniques, you can actually train models that not just integrate them, so put the data together, but actually translate between them. And so that means that you wouldn't in perpetuity have to measure everything for everything, but you would be able to measure one thing and actually learn the representation for a different one, or be able to map things in different areas. I think that would be actually the most likely path from a pragmatic point of view, because as Chloe said, there isn't one size that fits all. Unfortunately, there simply isn't yet the perfect technology that does everything that we want. But by covering with multiple technologies plus algorithms, we can actually get to there.
Terrific. I want to make sure we get to the last question in the Q&A, and I would encourage again the speakers to answer other questions in writing. So Mandeep had asked about the efforts that are being done within the HCA consortium to address challenges around access and the cost of single cell and spatial technologies. There are a number of things that we are trying to do, and this is work that's led by the Equity Working Group of HCA. Just to name a couple of different approaches, we have for a few years been running a series of training programs in countries of need, largely supported by the Chen Zuckerberg Initiative. That includes workshops in both experimental, these are typically week-long workshops in both experimental and computational techniques, where from 50 to 100 attendees will really get hands-on experience in both of those different legs. We've run those in Ghana, we've run them in Chile, and most recently in Thailand, but involving researchers from across the region. So it's not just focused on the one country, but really a regional thing. These have been hugely popular. There's an enormous demand. Typically, these will actually become oversubscribed within 24 to 48 hours. So an enormous demand for these types of opportunities. And we're currently speaking with a couple of funders about continuing that program and really hope to expand it in the years ahead. Another leg specifically about cost and other logistical challenges. We're just starting to speak with some of the suppliers who do recognize those challenges, particularly around things like import costs, distribution costs, technical support, and so forth. So we're also just beginning to work with some of the suppliers about trying to find ways to get around those challenges as well. So I would encourage anybody to reach out to the executive office. Again, we really want to know, even beyond cost, which is obviously a major factor, what some of the other challenges you're facing are, and work with you to brainstorm on solutions. Aviv, did you have more you wanted to say on that?
Yeah, I'll add 2 points. The first one is Mandeep is asking about saying that, you know, single cell sequencing and spatial technologies, access is limited and so on. I actually think spatial technologies open a lot more of the way than single cell genomics has, because spatial technologies, many of them can be applied to FFPE tissue. That tissue source is not just more available, it is also more stable. And as a result, you can separate the collection from the profiling. And so a regional center in Africa can handle a spatial profiling technology, but the samples could come from a broader geographical span in the continent. This is actually something that was highlighted by several of our speakers from Africa, from our regional, from our local network for Africa. In our last general meeting, there were some excellent talks. They showed fantastic data, and they really highlighted the fact that this is easier to do than the single cell genomics was, and that's a sample piece. I also would point out that actually in spatial technologies, while the cost per sample is higher, the cost per cell is a lot lower. You get a lot of cells in that one sample. So again, while that does not answer the challenge of the diversity, because for that we need a lot of individuals, at least one can see the path. And then finally, in Africa, there's been a greater progress now in genomics in general. A lot of initiatives, for example, centered around genetic variation in the African populations for genomic sequencing. And that could be another opportunity for HCA for partnering, because for the genetic studies, while already one does this, maybe there's also another opportunity with the same patient or individual base. So as John said, we're making a lot of effort from the central office, from the work together with the regional network in Africa, because in the end, all of this work happens there, and have actually hoped that technology will have a substantial deal in this as well.
Wonderful, thank you. In the few minutes we have left, I want to provide a few updates and a few links to resources, but I want to thank all of our speakers. All their presentations have been really wonderful. The questions have been fantastic. Really thank everybody in the audience for tuning in, for asking terrific questions, and for your interest and support for HCA. I'll just go through a few final updates in the last couple of minutes. If we can go to the next slide, you've heard about the biological networks and the regional networks, as well as the general meeting. These are all great avenues for you to participate in HCA, to lend your thoughts, your suggestions for future work, and to brainstorm again on what HCA's next steps should be. I would say if you would like to get involved, as with many things in HCA, I would say your first point of contact could be the executive office, and certainly we can put you in touch with the leaders of these various networks. I'll come back to the general meeting in a minute, so you'll have another chance to look at that QR code. Next slide, please. Here are some links for general inquiries. The email that we have, which is at the bottom of our homepage, is hca@humancellatlas.org. Specifically for inquiries about meetings, you can write to meetings@humancellatlas.org. And we have a help desk for the data portal, which is datahelp@humancellatlas.org. And again, you can find all these emails on our website. A few other resources. There are many resources that are available on our website. We have a page now with all of the policies and procedures that we've assembled for HCA. I encourage you to look at that as a first stop for answering your questions. We have a searchable database of publications that have been submitted to HCA and produced by HCA members. There are over 450 papers to date in that database. We also really encourage you, if you are preparing to publish a paper, to submit it to HCA for consideration. And we have a publication committee that will then review and approve your paper as an HCA publication. Information on that process is on that webpage. And I want to point out again the HCA Ethics Toolkit, where the ethics work group for HCA has generated and compiled a number of really helpful resources on consent templates and other aspects of the ethics of data collection and analysis. Next slide, please. A couple of upcoming releases. We are working hard, the executive office, on some updates to the website, and so you'll be seeing those. We're also very excited to be working on a brand new member portal, which beyond simply allowing us to maintain contact, will actually enable you to have your own profile on the HCA member portal, and we'll actually be able to connect with other members who are interested in forming new collaborations as well. A lot of other functionalities that will be built into this new member portal, so stay tuned in the weeks ahead for the release of that. Next slide, please. I also want to make sure that everybody is aware that we have agreements with 9 companies to provide discounts to HCA collaborating members. And by collaborating members, I mean the HCA members who have registered projects with HCA and are contributing in several different ways to the progress of the consortium. You see their identities here. If there are companies that are not listed here, I would encourage you to have them contact the executive office, because we are definitely always looking to sign up new members. Next slide, please. I would want to highlight the next HCA Town Hall, which will be Wednesday, the 26th of March, at the same time as this one. I encourage you as well to follow us on various social media, including Facebook, X, Blue Sky, LinkedIn, and YouTube, where we post all of the videos from all HCA meetings, including this one, which will be posted within probably, I would say, by early next week. And final slide, please. And then finally, I do want to encourage everyone to register, if you've not done so, for the next HCA general meeting, which will take place in June in Singapore. I'll point out that the early bird pricing ends on 8th of March, so I encourage you to register before then. You can use that QR code, or of course, information is on our website. Also, for those submitting poster abstracts, the abstract deadline will be the 28th of March. Again, I just want to emphasize that this is really a terrific way to engage with the HCA community and contribute to our future development. I hope all of you will attend, and look forward to seeing you in Singapore. So with that, I will close. Thank you all very much again to the speakers and to the audience, and we look forward to hearing from you in the weeks ahead. Thank you.